Welcome to Under the Microscope. This series is brought to you by the Real Scientists Nano team. Our goal is to provide a platform where scientists can communicate their work and interact with the public. With that in mind, every week we introduce you to a scientist working in the field of materials and nanoscience, who would be curating the RealSci_Nano Twitter account.
Hi everyone, today we have with us Andrew Kerr, who is a group leader and Chancellor's Research Fellow at the University of Technology, Sydney in Australia. Hi Andrew, how are you?
Hi Pranati, no, I'm good, thank you. Thank you for having me.
Looking forward to learning more about you as a scientist and your research as well. So let's start by understanding your scientific journey so far. So tell us about your scientific career.
Oh, where do I start? Well, I suppose if I wanted to go right back in time, I would say I was always interested in science as a kid. So I remember when I was, I'm going way back now, I remember when I was really young, I got a microscope for Christmas, and then I spent my whole Christmas outside collecting bugs and trying to look at them under a microscope. So that's probably where I really first started being a bit enthralled by the natural world and kind of going down the scientific route. So it started very early. So I did all the stuff in high school, and then I went to university. I'm from New Zealand originally. I'm a Kiwi. So we like our rugby and our pavlova. I went to a university called Waikato University. It's in the middle of the North Island in New Zealand, and I studied biological sciences. After I finished my bachelor's, I went and I actually started working for a vaccine production company for animal vaccines for sheep. And so I worked there doing the production facility down there. Got a bit bored of that. So then I went back to school, and I went back to Waikato Uni, and I did a Master's of Science in Biological Sciences. And I was looking at probiotics and the effect on the microbiome in the human gut. So it's a bit different. And then I went to Auckland University, where I was doing a bit more of that sort of work, looking at probiotics with the effect on the gut, and in particular with Fonterra, which is the largest dairy milk company in New Zealand. They were funding that research to see what sort of additives they could add into their milk products to help the human gut. And that was interesting for a while, but I have a short attention span. So then I left there and I went across the ditch to Australia, to Sydney, to do a PhD at Macquarie University, which is just to the north of the main CBD of Sydney. So I went up there, and it was actually to work on biofuels. So yeah, engineering fungi to produce biofuels. It was very different from what I had been doing. So I started to do that, and yeah, one thing led to another. Lots of things didn't work, and somehow I ended up looking at proteins that bind to silica, which is it was quite a big change in tact. Yeah, so it was these proteins bound to silica, and what we found is that we could actually then engineer those proteins to have another protein attached to them. So then we could attach lots of different proteins to the outside of silica. So that means we could functionalize the silica and give it lots of new functions. So we were mobilizing things like new enzymes to make pathways. We were also putting antibodies on the outside of silica nanoparticles that could then deliver silica nanoparticles to cancer cells and deliver some sort of therapy. So that's kind of where my interest in nanomedicine started coming into play. So when I finished my PhD, I actually went straight into a more nanomedicine kind of focused role, looking at nanoparticles and drug delivery. And that was with the ARC Center of Excellence for Nanoscale Biophotonics. It's a bit of a mouthful, but we call it the CNBP. So I worked in the CNBP as a postdoc. That was my first postdoc. And again, I just continued that, looking at delivering drugs with nanoparticles, but using proteins as the functional shell that attracted those particles to cancer cells and delivered the drug into them. And I really got into that. And because I was quite interested in proteins, that was my experience, and then nanomedicine, I kind of combined both of them and started looking at protein-based nanoparticles. So they're fully proteinaceous nanoparticles, and then using those for drug delivery. And that's where I got my first fellowship. So I got in 2018, I got a Cancer Institute New South Wales fellowship to explore re-engineering protein nanoparticles called encapsulins, and re-engineering them as drug delivery vehicles. And I've been doing that the last few years. And last year, I was lucky enough to get a Chancellor's Fellowship from the University of Technology, Sydney. And I've just moved my research group here, and we're just getting the lab set up. And it's an exciting new fresh start for us after 10 years at Macquarie. And we're really excited to be carrying out our research here now at UTS, which is it's in the city now. I'm in the city, I'm not outside of Sydney anymore. There's lots of good places to eat food.
That's definitely a plus, absolutely. But that's quite a journey you've had. You've worked at several different things. You have gathered experience from sheep vaccine production. Was that it?
Yep.
And with biofuels, with fungi, and all of it sounds so cool. So do you think now you have found something that is like really you're going to stick with for quite some time? Because you mentioned that your attention span is short-ish, sort of. Is there something what you're doing right now? And we'll get into that, the details of what you're doing right now in a bit. But yeah, do you feel like you found what you want to stick with for the next 5-10 years? I don't know.
Oh, that's quite a long time. Well, put it this way, I'm not bored yet, and I'm quite surprised.
Oh, okay, that's okay.
Yeah. No, I wake up in the morning very excited to be here, very excited to see things and what we're doing and playing around with these protein particles and making them do weird and funky things. And, I've got a great team that I work with, great collaborators that make it a lot of fun. So, I think over the last few years, we've been doing a lot of work, and now the papers, the publications are starting to get put together and put out now. So we're starting to have some output and some impact, which is really nice. When it all starts from a crazy idea and then you start having some outputs, it's good to contribute.
Right. It's nice to be part of that, like from conceptualization to actually putting it out there as a publication and sharing it with the world. I'm glad that you're still excited. You get up in the morning, you're excited to go in the lab and do the work. That sounds amazing. So you did mention a little bit about your current research in the short podcast, but where does your current research fit in this big picture of materials and nanoscience, which is quite broad?
Yeah, so I think we fit in that area of where we're re-engineering nature, so natural materials. So we're taking something that already exists in nature and then using genetic engineering and chemical engineering to change it for a new purpose. So I think we're definitely in that natural material area. That's where we are ourselves.
Okay, like you mentioned in the short podcast that you teach proteins to do other than their everyday job. Was that it? Was that the right way of saying it?
Yeah, that's a bit of my catch line at the moment. Yeah, so what we do is we take proteins away from their normal day jobs and we give them a new job to do something else. So so in our case, our basic research, we're basically taking protein-based organelles. So these are little protein-based nanoparticles called encapsulins. They're found inside bacteria, and they encapsulate enzymes that allows them to then function like a little nanoreactor to do different jobs. So and usually it's to help the cell overcome sort of stress, like oxidative stress or something like that. So they're an organelle that helps the cell, the bacteria. And what we do is we can take that out, or we take the gene that encodes that protein inside that bacteria, and then we just put it into another workhorse bacteria and start making it in the lab. And we can change that gene, we can engineer it to have a different property. So then when we make the nanoparticle, it has a different function. So that's kind of what we're doing. Yeah. So it's still a
Okay.
protein material, but we just manipulate it.
That sounds so cool. That just sounds so cool. It almost makes me want to go back in the lab and go back to my past life as a scientist. It sounds really, really
It's
cool.
never too late.
That is also true. Yes, I remember that. Uh, so Andrew, it sounds that you, not just your current research, but also in the past, you have been involved in a lot of interesting research projects or experiments. And this is a difficult question. I know you're asking it as well. It's a difficult question because I'm going to ask you, which research project are you most proud of? Because I'm sure you have a bunch of them. You're proud of most of your research projects. But if you have to pick one, could you pick one and explain it to us in simple words in the section we call In Other Words?
Yeah, it is a good question. I'm usually proud of anything I actually finish.
Ooh, okay.
Yeah, because that's always hard. Finishing something. Starting something's very easy, right?
Right.
So I think like recently, I'm really proud of our latest paper, our latest research article that came out in ACS Applied Materials and Interfaces in February. So this is a paper that kind of represents a few years' work by my former PhD student Denise Diaz, and it's where we first really started playing around with these protein nanoparticles, and we managed to engineer them for photodynamic therapy. So basically what we did is we took these, using genetic engineering, we took these protein nanoparticles out of bacteria, and we engineered them to encapsulate a protein called a photosensitizer. So this protein photosensitizer was inside the nanocage. And what we were able to do then is shine a light on that little nanocage, and it converted oxygen in solution into reactive oxygen species, or ROS, which is actually quite toxic to cells. So then what we did was we delivered this engineered nanocage into a lung cancer cell. We shined a light on the lung cancer cell. That cage started to generate that ROS, and it started to kill the cell. So that's the photodynamic therapy. So we managed to do something really fun, really funky, and yeah, we actually had a little application there, and it was exciting. And hopefully we can carry that work on further.
That sounds really, really cool. I have a very silly question, probably. Is there like a particular wavelength of light you need for this to turn this oxygen into the killer version of oxygen, if that's the right way of saying it?
Yeah, so in this instance, the protein photosensitizer that we encapsulated inside the particle, that's activated by blue light.
The blue, the
So
five.
activated by blue light to generate reactive oxygen species. So the problem
Right.
there is that blue light doesn't penetrate through tissue very well at all.
Yeah.
So it's more conceptual in the sense that you could then move to a different protein photosensitizer and put that inside the cage instead. That has a longer wavelength and then more penetration. Alternatively, we worked at the time, in the CMBP that I talked about earlier, they generate a lot of fibers, optical fibers that can deliver light into weird and strange places in the body. So we can use that as an excitation source if we're worried about a lack of penetration with the light.
Okay.
Yeah.
Okay. Wow, that sounds so cool. I'm definitely going to read that paper. Definitely going to put the link in the description as well so that everyone can read that paper because it sounds so cool.
Feel free to cite it.
Yes, people, listeners, feel free to cite the paper as well and talk about it on all social media platforms of how cool the paper is, and tag Andrew when you're doing that, right?
Sounds good. Sounds good to me. I sent it to my mum and dad, but I don't think they were. I don't think they read all of it.
Yeah, I had similar experience with my parents. They have my theses at home, back home. They're like, yeah, we have them. I'm like, have you read them? Like, yeah, we have them. Have you read them? No, we have them. We are keeping them. So they're just dodging the question. Um, but I'm sure they are proud of you for doing such amazing
Just like
research.
yours, I'm sure, definitely.
Yes. So it's clear to me, Andrew, that you love the research aspect of being a scientist. You do, right? You love the research aspect. You love going in the lab probably and doing the experiments and data analysis. I'm assuming that.
Oh yeah, I love all the aspects of being a scientist, except maybe the admin. But other than that, like all of it is fantastic. It's great. Talking to you today, that's part of it all. Yeah, love it.
That's great. That's great. So, other than the research part, what else do you like about being a scientist? Because oftentimes people don't know what else um, keeps people in being a scientist, basically.
Yeah, creativity and imagination. Like
Okay.
we have the opportunity to come up with crazy ideas and be truly imaginative. Like and then get to explore those ideas. It's a unique privilege that a scientist has
Mm-hmm.
to explore ideas in the depth that we get to explore an idea.
Mm-hmm.
Yeah. And sometimes it can be quite self-indulgent, I think, but like it is an amazing thing that I think, yeah, that we can't take for granted. Yeah.
Absolutely. So, creativity and imagination.
And
That
the ability to explore those ideas and yeah, that's
also that. Yeah.
yeah.
I,
So I think
I think
that's the most amazing thing about being a scientist.
I agree. I think one of our curators in the past also said that being a scientist is like being an artist. You have it in your head. You have the piece in your head first, and then you conceptualize it. You create it. You imagine it first, and then you create it, and then you actually work on it, make it happen. And if you're lucky, then everything works fine. And then there is a publication about killing tumor cells with the nanocages. Was that correct?
Yep.
So yeah, I agree. I'm glad you see it that way.
It's so
Um,
true. Like, yeah, you really are like an artist, right? And you've got a blank canvas, and then, yeah, and it doesn't always translate the way you want, right? The picture doesn't always come out like it was in your head, but, like it's the journey.
Absolutely,
Yeah.
absolutely, absolutely. Oh, I'm so happy. I'm really considering going back to being a scientist now. Uh,
Or an artist. It's the same thing.
Yeah, it's the same thing. Yeah, it's
Yeah.
it's totally it's the same thing. Absolutely. So Andrew, you have quite a lot of experience so far as being a scientist, and you also have a group um, that you're leading. So, what advice would you give yourself or the scientists that are starting out right now, if you were to go back in time and to start off, like start your scientific journey? What advice would you give?
Well, yeah, that's another good question. um, so if I
You have
could
a
go
bunch
back
of
and
advices coming to you, aren't they?
yeah, I mean, if I could get back in a time machine and go talk to young Andrew,
Mm.
I'd probably whip out the old saying, "Fake it till you make it." It's just be confident. Be confident. Talk to people. Don't be afraid to talk about your research. Don't be afraid to talk to others about their research. Put yourself out there.
Mhm.
Like put yourself out there. And like when I say fake it, it can be that you can be so nervous and you can be really shy and
Mhm.
reluctant to talk to someone, especially if it's a big professor in the research area you enjoy. Um,
Mhm.
but sometimes you need to fake that confidence to break down that first wall and then start talking to someone, and that'll start opening up doors for you.
Mhm.
And so that's the big thing. So fake it to make it. Fake that confidence. Get out there, and eventually it will come. It will come, and you'll just won't be able to shut up, and you'll be talking to everyone and collaborating everywhere.
Okay. Okay, that's some really good advice. So it's basically don't let the imposter syndrome stop you from talking to people and communicating. Is that
Yeah,
a
yeah,
different way of saying it?
yeah. That's right. Yeah, yeah. So
Yeah.
just
All right.
put on that, do the Superman pose. You know, like people say, you know, before you go to give a big talk, you do the Superman pose.
It
You know,
works, you know? It
yeah,
does work.
yeah. I won't lie, I've done it before. It works. Just gives you
It's
that real feeling of like confidence, and then you get out there and you just once you get started, gets easy.
yeah. It's just that getting started, that you just need that little push. So either fake it till you make it, do the Superman pose, whatever you need to do, just do it. That's your advice then.
Yeah.
That's some really good advice. Awesome. So I hope your Andrew, your research experience has been wonderful so far. I mean, you are still doing research, and it will continue to be amazing also in the future. But if you had 3 wishes to improve your research experience, what would you ask for? And I'm not promising anything here. Sorry.
um, more money, more money, more money.
Um,
I think, well, obviously, having a bit more cash to do research. I think everyone wants to have more funding, and it's fiercely competitive out there for research funds, no matter where you are. So that always makes life a bit easier to be able to explore a few more ideas if you have a bit more cash. It'd be nice. I guess wish number 2 would be I'd like a few things to work the first time I do them. That's it's a rare event, but when it does happen, you enjoy it. If that happened a bit more often, life would research would be a bit easier, and we'd be moving along a bit quicker. And I guess I would just wish to have even more great collaborators and people that I work with. I enjoy them, and I enjoy collaborating. I have some fantastic people that I work with across Australia and internationally, and like, yeah, just more of those. That would be another great wish, research wish.
Okay. All right. So more money, things to work the first time so that
Yeah,
the journey is quicker and more fun and more enjoyable, so that you can also move on to the next project, right? So that you're not stuck with trying to fix the process. You can use the same time to work on different ideas. That's the reason why you want the things to work the first time, right? The second
I think
wish.
so. No one likes optimizing.
Yes, it just should work. I have it and it worked in my head. Why isn't it working in the lab? I
Yeah,
understand. I
it's the
understand.
painting. My painting's not coming out as nice as it was in my head. The painting looked good, but yeah,
Yeah, yeah. Why do I have to do it 10 times to make it better? Just first time because I have 50 more ideas in my head that need to be worked on. So
yeah.
I understand totally. And also the collaborations rather. All 3 very, very understandable and relatable wishes. I hope they all come true. I hope you have more success for the first time trying new things. Unfortunately, I can't grant the wishes. I'm not Thanos to snap the fingers at me like that's how it's going to be. But I would like to
Oh,
believe
I wouldn't have signed up if I knew that.
that. I might have to tell you something else later. No, I'm kidding. Uh, but no, all 3 really good wishes, and I'm sure you'll make it happen. At least the collaborations part, that is definitely in your hand to a certain extent. So I'm sure that will work out. So Andrew, this has been wonderful. But before I let you go, I have one last question for you about the for lack of a better word, an interesting year that we all lived through, which was 2020, and we are still kind of living through it. We learned several things, we unlearned quite a bunch of things. So, would you like to share some of your learnings from the year 2020 with us?
Yeah, it's been a challenge for everyone, hasn't it?
Hmm.
Some more than others, that's for sure. Very lucky
Hmm.
to be in Sydney. Very fortunate in Australia.
Hmm.
I think one of the interesting things that I learned was that we're very interested in having supplies of toilet paper in an emergency. I didn't expect us to be so concerned with that, but that's what I learned. But on a more professional aspect, I think really learned more about communicating clearly and keeping positive. Like, I think that those are the really important things. So like communicating regularly with people in your team, understanding how they're feeling, being empathetic, and just being open. You can be open about if things aren't going well, or like personally or not. Professionally, same thing. Just, yeah, communication is so important, and everyone needs someone to talk to, right? Like, people need each other. So I think that's what I learned about 2020 is like, yeah, we can all be a bit better and be there for each other.
Okay, yeah, absolutely. Be more empathetic and just listen every once in a while if you can. Yeah, communicating. That's a good point. And the toilet paper, of course.
Yeah.
That complete different side of how humans work and think in a pandemic. Like, really?
Yeah.
Toilet paper. But yeah, well, as I said, this has been wonderful, Andrew. Very, very excited to have you on Real Scientist Nano. I'm definitely going to follow all your tweets and the GIFs and the pictures and everything that you're going to tweet about. So thank you very much for speaking with me.
No, thank you for having me, Pranati. I'm looking really forward to it. Thank you.
Thank you for listening. To know more about us, please visit our website, realscientistsnano.org, and follow us on Twitter at realsci_nano.